Thank you. So yeah, as you just said, so I'm Aurélien Luciani. Today I'll talk about UniProt, the Universal Protein Resource. So UniProt is made by a consortium composed of SIP in Switzerland, PIR in the US, and Amboli BI in the UK, and that's where I'm based, in Amboli BI. So first of all, what is UniProt? So the mission of UniProt is to support biomedical research by providing a rich resource of freely available, stable, comprehensive, and accurately annotated protein sequence and functional annotation. What does that mean? It means that we gather all the sequences that we can find in the literature or in other databases and try to have one place where you can catalog all of them. So all those sequences end up forever in our database, in our archive of sequences, which is UniPark. And then some of those sequences get brought into the UniProt knowledge base or UniProt KB. And so on top of the sequences, then we add some functional annotations, subcellular locations, pathways, catalytic activities, all of this information that you might be used to find in the UniProt website. We also have structural information, so 3D structures or predictions of 3D structures. And on top of all of that, we also link that to different tools. Mostly known are BLAST and the Align tool, which is based on Coastal Omega, but we also have a peptide search feature and a ID mapping where we can map UniProt information from and to other databases. If you have a pipeline that requires that, that's quite useful. I want to highlight that UniProt is a Elixir core data resource, and it's also a global core biodata resource. And here you can see on this tile here that the UniProt knowledge base or UniProt KB, we have about 245 million entries in there. We have it's split between the reviewed one, also known as SwissProt, and the unreviewed one, also known as Trambo. And so the reviewed one are reviewed by humans, by curators that will annotate those. And the unreviewed one will be annotated through automatic annotation. So as I said, the UniProt website is composed of UniPark, the archive of sequences. There's a UniProt knowledge base here, and we also have UniRef, which are clusters of similar proteins by identity, and also proteomes. And so proteomes are groups of protein expressed by a organism and grouped by assembly. So we have 640,000 different proteomes in there, and you will be able to find all of those through the UniProt website. So if you've started using UniProt websites in the last 2 years, this is what you've always known, but this is actually a new website that we've introduced in July 2022. So this new website, the goal is always to have the open access, unlimited and worldwide, but we keep having more data and more users, so we need to update a bit the architecture to be able to cope with the new information and the new data. So what we have in a new design and improved architecture with modular setup so that hopefully it's more stable and it's able to cope with more users than ever. We also try to find a balance of design for recurring users versus casual users. So people might be going every day for the last 10 years to UniProt. We still need to be able to cater for those use cases, but we also want to be welcoming for new people, for people that don't usually use UniProt, so that they can find the information they need in the best way possible. And we also have decoupled data and website API releases. So the data, some of you might know that it's only released every 8 weeks, and the website and API can be updated at any time. So if we need to do a bug fix or add a new feature, we're able to do that in 20 minutes from the time we fix it to the time it's actually available to users. One new thing that we added in the new website is a tool dashboard. Before, we didn't have a dashboard, so people submitting a job, so for example, a blast job or an alignment job, they would need to keep the tab open or bookmark the results page until the job is finished. So now if you click on the top right of the website, you will have, coming from the right, a tools dashboard where you can find all the jobs that you previously run that are currently running. And with all of this information here, you can also customize title. If you submit a lot of jobs, that could be useful, because otherwise, I mean, you would be seeing 5 different blasts and you don't know which one is which. So you can customize all of this, and you have also some extra information about the job itself. It allows you to rerun the job, and you can modify it when you rerun it if you want to adjust the input parameters and all of that. So the jobs are, as before, kept for 7 days, but now you're also able to keep the job details for 14 days. So even when the job expired, you're able to rerun it from the same input that you had in the past, and you don't need to remember that. And if you want to mark it because, I don't know, you want to rerun a blast job after every release, you can mark it with a star there, and it will be kept forever. And the point is that you can continue browsing the website. You can even close your computer and come back later. It will be kept here. We added some visualizations in the new website. So you might have seen already the Prot Vista feature viewer in the previous website. It was already there, but now we also cut it to pieces and put them across the UniProt website in smaller bits. So here, there's a lot of information here. It's all the metadata, all the annotation that we have for a specific protein. On the x-axis, you have the sequence. And then you have groups of different features that we annotate there. But also, if you browse the website, you will be able to see like mini versions of it. Here we see only the site's information, for example, and you will see that across the website. And the fact that it's modular and reusable in different places also means that is something that we've developed in collaboration with other teams, and they're also using the same visualizations in their own customized way. So there is PDB, Interpro, Open Targets. They're all using this in some way. And also, if you're interested in using that, you should be able to use that. So get in touch afterwards, and I can tell you how to do that. So this is based on a library called Nightingale. We also integrated some other third-party or collaborators visualization. So here's an example: is the Go ribbon showing genome ontology annotations. Gene ontology annotations from the Go ribbon. We also added the subcellular visualization from SIB, where you can see where a protein is in the cell. You also have the react catalytic activity, so where you can see this kind of information, also using a visualization from another team. And the last one to be added just last week is the complex viewer. This is developed by a complex portal at EBI, and now we're able to see for a specific protein all the interactors and where in the sequence it interacts with other proteins. And so upcoming, we'll have other visualization that we'll be adding, so GoCam and Expression Atlas, for example. We also updated the API. So a few things I want to highlight is, for example, the new format that we added for embeddings. So that way you can also directly download from our API embeddings. You don't need to generate them. And there are some of them that are pre-computed, for example, for the whole human set of proteins, but you can also generate them on the fly for any query that you do on the website. We also expanded the TSV format to have a full option. So before it was only returning primary identifiers for some cross-references, but now there's this option to actually return more secondary identifiers. So it's quite specific to the cross-reference that you're querying, but now you have access to more information than before. And we have different download options. Depending on your use case and what you want to download, one might be more suited than the other. So I've highlighted here the preferred use case and the fact that it's resumable or customizable. And then based on that, you can decide which way is the best. So all of this is documented on the website in the help pages. Just want to highlight that taking advantage of the job dashboard, we introduced the API file generation. So that's the fourth column here. And basically, it allows ask the website to generate a file, entirely customizable, and then come back to it later. So the website will be doing its work in the background, and then you will be notified through the job dashboard that your file is ready to download. So that way you don't need to keep the website open or to keep your browser open or to run a script. You just come back later and your file will be ready for you. I want to note that what you see on the website, everything is downloadable. It's coming either from the core UniProt data through the UniProt API, from the Proteins API, which is more information mapped to UniProt, and also third-party APIs. So you will see on the website there's some download buttons sprinkled all over the place, and you should be able to download everything that you see there, and also to get the URL that you would need for programmatic access. We added, since the last 2 years, more cross-references to other databases. The biggest one is AlphaFoldDB. but we also added AGR, Glycosmos, Pumba, EMDB, and Japonicus DB. And there's been a change of name from Tiger Farms to NCBI Farm. So these are only a few cross-references, but we actually have 189 cross-references that you can configure and download from our website. And this is just a part of the 384 fields that you can customize. So you can decide to view these on the website or download them. And all of this is customizable. We also added some other information through, in that case, the proteins API. You will have access to genomic coordinates. So for a specific protein, you will be able to map to the corresponding position in the genome. And you will have here links to, in that case, Ensemble and the Ensemble genome viewer, genome browser. And you will see that in this example, this protein is expressed from, well, the chromosome 21 at the specific position. That's for the whole protein, but you also have the level of information up to the exon. So you will see which bit of the protein is coming from which exon, and you will have all of that information for each of the isoforms that we have on the website. We also added PTMs, antigens, and epitopes. And so, yes, we keep mapping more and more information to UniProt, and this is all available through the Proteins API provided by UniProt. Some of them are already embedded in the feature video that I've shown you earlier. So in that case, we're viewing PTMs, and the others will be added soon. We also added AlphaFold and AlphaMissense data. So AlphaFold has been there since the day one of the release of the public release of AlphaFold. And if or when there's a new release of AlphaFold, we will have that immediately on the website. So the point of viewing that through the UniProt website is that you can see this prediction and in context of all the other annotations that we have. So here I'm playing with the domains or region information, and I can see where it maps to in the 3D prediction from AlphaFold. And since last month, we also added AlphaMissense data. So AlphaMissense will allow you to view pathogenicity information, prediction of it at least, for all the possible mutations, for all the possible positions in the protein. That is on top of the existing variant information that we do have in the website already. To stay more on the AI ML stuff, I want to highlight our collaboration with Google DeepMind, where we've been able to add the protein method to add predicted names for previously uncharacterized proteins. So that's 28 million of them. And so this is a method based on sequence and taxonomy, and that will output on the other side a protein name. And we're also working with them to develop more methods to be able to predict more different annotations. I want to also highlight that tomorrow there will be a talk by Vishal here, that will talk about what we do in the UniProt team regarding LLMs and other related work. This was a lot of things in 10-15 minutes. I want to tell you that there's actually a lot of webinars that we have on the Humble EBI training website that's been recorded by myself and other people in the team. So you're able to have 1-hour webinars about how to access the API with using Python, for example, or a walkthrough of the new website. This is all available here, and yes, I encourage you to go there and explore more of the training resources that we've developed for all of you to use. So I've shown you a lot of things that we've been doing, a lot of things that we will be doing, and me being here is also the occasion for you, if you're a user of UniProt, to tell me what you want to see next in UniProt. So do you want to add a new visualization to it, new XREFs, or new methods, or you want some improvements? Come talk to me at some point during the conference. I'll be around. And really, every new development that we do is based on the user feedback. And so that's also why I'm here today, to be able to get the feedback from all of you. And to finish, so I want to highlight the work that's been done by all of these people in the consortium. As you've seen, it's people at the EBI, at PIR, and at SIB. And that's a lot of work that's been done by all of them, curators, developers, production team. There's a lot of work behind the scene for you to be able to access all of this data. And obviously the funding, I want to thank the funders for this. And finally, I want to thank you and take the opportunity to listen to any question that you might have. Thanks.
Great
Hi.
talk. So I use UniProt a lot, and sometimes I wonder for the localization, there's a lot of localization databases that are out there, like SPC scores and such. Do you consider adding that into UniProt to your like localization? Oh, sorry. Just to further highlight the localization of the protein.
So is that cross-references that are already cross-referenced in the website, but just not visualized, or is it new ones?
It's new ones.
Yeah.
And I'm happy to talk to you about it, but
Yeah.
those are things that we use in our lab to make sure that, we actually we're confident of the localization using different methods. But
So yeah, I can take note of that. So, but usually to add new cross-references to the website, there would be a set of requirements that would be evaluated by the team. So for example, that would be the coverage of proteins. For example, if you have a database that covers only 10 proteins, it's unlikely that we're gonna add it because I mean, there's a lot of things that we need to maintain, and that would be, but if it covers a significant amount of a proportion of UniProt, that's a good candidate. Also, if it's updated regularly, that's a good candidate.
Mm-hmm.
And that would be, if it's not something that's already covered by other cross-references, that would be also a good candidate. And that's evaluated by the team. So I'd be really happy to hear from you, like the specific list of them.
Sure, definitely. Thank you.
Thanks. Hi.
I was just wondering about sort of the difference or overlap with Magnify proteins and UniProt, and like sort of how those two things are growing and where you see them going together.
With Magnify?
Yeah.
Yeah. So it's another team at EDI. I'm not sure really, we don't really have much overlap there. I don't think we have the cross-reference yet, but we are planning on adding more cross-reference between the different resources. So yeah, at the moment it's different work made by different team. Yeah, there's nothing really that I can tell you at that point. Yeah,
Great.
sorry.
Thank you.